Hey guys, it's technically Friday for y'all, so really excited. We have made it to yet another weekend. I'll pull up the remote learning guide, and we are right here with the Punnett Square problems. Today we're going to talk about chromosomes, Punnett squares, stuff like that. If you've done Punnett squares, it will probably have been in biology. Hopefully you remember it. If not, we're going to do a little bit of practice today to kind of help you to remember what that is. If we look down at the next, what we got going on for next week, we only have 2 assignments, Tuesday and Thursday, and nothing. What does that mean? That means you guys are done. You guys are less than a week away with being completely done with Animal Science 1. You guys will have made it. Big congrats. Pulling up the PowerPoint though, we're going to get into today's lesson. So like I said, this should be like relatively a review from biology. Chromosomes, they're little rod-shaped bodies. When we typically talk about them, we talk about them being in pairs. And they're the same. Cattle have 30 pairs of chromosomes. Pigs have 19. Chickens have 39 pairs. How many pairs of chromosomes and how similar the DNA is determines what animals are able to mate. So that's why we can't have pig-cow combos. They don't have the same number of chromosomes. They don't have the same number of pairs, meaning they cannot breed. This is what determines what animals can breed and what animals can't for when we have hybrids. So I always think that's really interesting. You can't just like put all these animals together and have a 50-50 mix, because even if they were, by some crazy reason, able to mate, there will be way more cow DNA in there than hog DNA, because cows have 30 pairs of chromosomes and hogs or pigs only have 19. So different breeds of cow, they all have the same number of chromosome pairs. They all have 30 pairs, which is how we can get like a Hereford and an Angus cross. They're different breeds, but they're still the same species. They're still cows. They still have the same number of chromosome pairs. Chromosomes, they're made up of DNA. They're found in the nucleus. Before like meiosis and mitosis, the DNA is just kind of like floating around in the nucleus, doing its own little thing. And when it comes time to like essentially reproduce, to go through mitosis or meiosis, that's when the DNA starts forming into the actual chromosomes. Genes are slightly different, so that's why I have this little graphic down here at the bottom. I feel like this makes it really easy to understand, because genes and chromosomes sound very, very similar. Not like sound similar, but they perform essentially the same functions. So this graphic helps you to see where everything is when we talk about things like DNA. We've got our cell down here, and we've got our nucleus. Inside we have our chromosomes. We've got our pairs. Then if we look at the entire DNA strands, the chromosomes are all in here. Tons and tons of chromosomes make up genes, and a bunch of genes make up DNA. That's why I love this graphic. I feel like it makes it so much easier to explain. Rather than me telling you that genes are a bunch of chromosomes, you can look and see in the graphic how the size proportions and how everything kind of relates to each other. Genes are located on chromosomes, and they control all the different characteristics that an animal is going to inherit from its parents. There's thousands of genes in every animal, all different kinds of traits. What color is your hair? What style is your hair? Is it straight? Is it curly? Do you have freckles? There's a gene for that. Do you, trying to think, what's another one? For guys, like, how much facial hair are you gonna grow? Are you gonna be able to grow like a really big beard? Or are you not gonna be able to grow a very big beard? Are you gonna have a lot of hair? Or is your hair gonna be really, really thin? What color are your eyes gonna be? There's all different kinds of genes. When we talk about animals, though, the primary genes and characteristics that we want to talk about are things that make the animals better for us. So different carcass traits. Sorry, there's a big truck driving by. Different carcass traits, like marbling, like the ribeye area, things like that. Growth rates, how quickly does the animal grow? Because the quicker the animal grows, the less we're really going to have to feed the animal. Feed efficiency is also really important. If you can feed two animals the same amount of food, but one grows more quickly because it's able to use the food better, then those are the animals that we want. We don't want the animal that we have to feed more and more because it's just not necessarily using the food the way that we want it to be. So there's characteristics, there's genes for all of these different things. This is why breeding is so important in animals. You don't really think about it, but I mean, there's just about a gene for everything. There's dominant genes and there's recessive genes. A dominant gene is when we look at something, when we look at a cow, that's what we see. Dominant genes, they're dominant. They're going to cover up or hide the recessive traits. Wow, my neighbor's dog's very loud. So if we want to talk about an example, cattle being polled, meaning they're born without horns. This is something that we look for. This is something that we try to breed into our animals because if they don't have horns, then it's less for us to have to necessarily worry about. We're not going to be worried about them fighting for dominance with their really hard horns. We're not going to worry about their horns getting stuck in something because that happens a lot. So we've bred polledness into cattle. We pick the cows that are born without horns, and so that we can get more cows born without horns. This is a dominant trait, a dominant gene, meaning if you were to breed a cow, a polled Hereford, a Hereford cow without horns, to a horned Hereford, a Hereford with horns, then when they breed, the odds are very high that the polled Hereford, the cow born without horns, when they have a baby, that baby is more than likely not going to have horns because that's the dominant trait. There are recessive traits though. Recessive traits, they can cover up dominant traits, but they have to come in homozygous pairs. If you have a dominant trait mixed with a homozygous trait, so if you mix a dominant with a recessive, a cow without horns and a cow without horns, more than likely the baby they're going to have is going to show the dominant trait. They're not going to have horns. But if you breed two cows with horns, if you breed two not polled Herefords, two Hereford cows with horns, then the baby is going to have horns. So the way we try to talk about this and figure out like what their genes are, is with Punnett squares. So when we talk about these genes, we talk about them, they come in pairs. So it comes in 2 letters. In this example, P. In this example, Y. You can use just about any letter that you want to represent whatever the gene is that you're talking about. If we say that, excuse me. If we say that genes are homozygous, then that means that they're the same case. They're both uppercase or they're both lowercase. If the letter is uppercase, then that means we're going to say it's dominant. And if it's lowercase, then we're going to say it's recessive. Dominant uppercase, recessive lowercase. If it's homozygous, then you're going to have 2 uppercase or 2 lowercase. If it's heterozygous, hetero meaning not the same, so homo means the same. You're going to have the same case of letters, both uppercase or both lowercase. Heterozygous, hetero means not the same. So if they're not the same, then you're going to have one that is uppercase and one that is lowercase. One that has a dominant trait, one that has a recessive trait. So if we look at this example, this is how you set up a Punnett square. This example right here is for string beans. Sometimes the shell is green, sometimes the shell is yellow. And the color that the shell is depends on the genes. Because this is an uppercase Y, this is telling us that it is dominant. Uppercase means it is dominant. This is the trait that we should be seeing. And this is a lowercase y, that means that it is recessive. So if we have a yellow string bean, then it has at least one dominant allele, one dominant letter. If it's green, then it's recessive, meaning it has to have 2 lowercase or recessive letters or alleles. So when you mix them, you put one parent here and one parent there. So this person said, okay, if we have a yellow string bean and a green string bean, what are the odds? What are the babies going to look like? Inside each square, this tells you what each baby more than likely will look like. It's not a guarantee, but more than likely what it's going to look like. So you put one letter here for this parent and then the other letter on the bottom row. And then you do one letter here for the other parent and one letter here for the last letter of that parent. And then you just move them across the board. This yellow Y, this yellow uppercase Y goes here and it goes here. It goes straight across the board. And then the lowercase y, the recessive y that this yellow string bean has, goes across the board. It goes right here and it goes right there. And then we've got this parent. Both alleles, both letters are lowercase. This is a recessive trait. So little y goes here and little y goes here. And then the little y goes here and the little y goes here. And what you're left with is what the gene should look like in each offspring, in each baby. So you have the uppercase y and a lowercase y. If you have one uppercase letter, it's dominant, automatic. So this string bean is going to be yellow. Come down here, no uppercase y. Both of them are the little lowercase y's. This is a homozygous recessive pair. Homozygous meaning they are the same case. They're both lowercase. Recessive meaning that they're both not the dominant trait. So this string bean is going to be green. You come up here, same deal as the last situation. All it takes is one uppercase letter. This string bean is going to be yellow. This big Y covers up any trait that this little y has. And then we have another homozygous recessive pair, meaning this string bean is going to be green. So we already kind of went over this. The dominant gene is going to be written by an uppercase letter, a capital letter. And the recessive gene is going to be written by a lowercase letter. So sometimes, this is just kind of like a side note, there is such a thing as codominance, meaning that sometimes some genes have more than one recessive or more than one dominant trait. It doesn't happen too often, but the most common scenario where we see it is where wheat is in a coat color called roan, R-O-A-N, roan. Roan is a combination of red and white, meaning a red cow bred with a white cow, and the offspring has both red and white hair. Normally it's one or the other. You have a red cow or you have a white cow. But when you have a gene that is codominant, you see both colors. You see red and you see white. It doesn't happen too often. And if you were to ever see it, like in a problem or something, it would say explicitly there, like, these genes are codominant. If it doesn't say anything about codominance, then you shouldn't assume that anything is. So homozygous and heterozygous. A homozygous gene pair, homo means the same. So when we're talking about genes and we're talking about things being the same or homozygous, that means that the letter case is going to be the same. They're both going to be uppercase, they're both going to be lowercase. So a polled cow, excuse me. If we're talking about polled cattle, then homozygous is going to be both. It's going to be both uppercase for a polled cow. Or if we have a non-polled cow, it can still be homozygous, but it's not going to be with uppercase. It's going to be with lowercase, and you're going to have two little p's. And that's going to mean that that cow has horns. If the gene pairs aren't exactly the same, if they're not both uppercase or both lowercase, then they're heterozygous, meaning different. One trait is dominant, hence the upper or capital P, and then one is recessive or lowercase p. This big P is gonna cover up the little p though, because that's how dominant traits work. If for you to see a recessive trait, you have to have two of those recessive genes. If you just have one, that one's gonna get hidden. So when we talk about the P's and the Y's and stuff like that, we're talking, excuse me again, oh my goodness. When we talk about the P's and the Y's, we're talking about the genotype. There's my mouse, good. Hello mouse, okay. When we talk about the genotype, it's the kind of pairs. It's the DNA. It's whatever it codes for. So when we did that Punnett Square right there with the Y's, the yellow Y and the green Y, the uppercase Y and the lowercase y, we were talking about what letters we were going to have for us to see a yellow string bean or a green string bean. The genotype are the letters that we use to talk about what the colors are going to be. The colors that we actually see, or the physical appearance of the animal or the plant, so whether or not we got that yellow string bean or green string bean, is the phenotype. The phenotype is the physical appearance. So the genotype is when we're doing letters. The phenotype is what we actually see. So uppercase Y, lowercase y meant that we were going to see a yellow string bean. Two little y's meant that we were going to see a green string bean. The two little y's are the genotype. The fact that the string bean is green is the phenotype. Punnett Squares, they use genotypes to try to predict the phenotype. So here's an example. We got some octopus here. We have two parent octopus and their 4 little babies. So we have a green octopus and a purple octopus. And we see that their babies right here are all green. So we're going to walk through the steps on how to create a Punnett square. The first step is to figure out the genotypes of the parents. So we need to pick a letter, first of all. So we're just gonna go with A. Let's do it. Let's just do A. So when you're trying to figure out what is the dominant trait and what is the recessive trait between the parents, you have to look at the offspring. Looking at the offspring will tell you what trait they got from their parents. If all of these octopus were purple, that would be the dominant trait. But they're all green. So that means that the dominant trait here is to have a green octopus. So if the dominant trait, if we see green octopus, it has to have a dominant letter there. It has to have a capital A. That means that we know that that means that this purple octopus has to be little a, has to be recessive. The only way you're going to see a purple octopus is to have a recessive trait. So that means homozygous recessive, 2 little a's for our purple octopus. And then our green parent octopus, you have 2 options. We know they have to have at least 1 capital letter, 1 big A in there. At least 1 big A to cover up the purple parent's color. So we can either be big A, little a, or 2 big A's. So now the next step is to figure out the possible offspring the parent could have. So for every possible genotype, we need to try to figure out what the offspring would look like. That means we're going to have to make Punnett squares for each possibility. We're going to make a Punnett square for if the parent was big A, little a, or big A, big A. So here's what our Punnett squares would look like. We have big A, little a for green parent, and big A for green parent. This right here doesn't change because it's homozygous recessive. Homozygous recessive is the only way that we're going to see our purple octopus here. So this one isn't going to change. So now we cross them. Our big A comes here and goes there. Our little a goes there and there. And then we've got our recessive parent, which is all little a. So little a there, little a there. Come down here, little a there, little a there. And then you do the same thing over here. Then you look at what should these babies look like after you complete the Punnett square. Try to figure out which Punnett square is correct. So we look at, so we have a big A here. That means that we're going to have at least 1 green octopus baby. And we're going to have another green octopus baby. But then we get right here and we have 2 little a's. This is homozygous recessive, meaning we're going to see the not dominant trait. This means that we're going to have 2 purple octopus babies. Do we have 2 octopus babies that are purple? No, all of them are green. So this one's probably not right. But let's double check by confirming by looking at this one. So we have big A, little a, big A, little a, big A, little a, big A, little a, meaning they have at least 1 dominant green colored trait in here. So that means that all of these babies should be green. Are all of our babies here green? Yes, they are. So, like I said, we're just confirming that we're correct. We know that the Punnett square on the left can't be correct because half of the offspring would be recessive, meaning they would be purple, and they are not. So we know that that one is wrong, and this one is correct. Here is another example where we talk about polled cattle again. a dam is a mother. D-A-M is the mother animal. And a sire is the male father animal. So in our example, two polled cattle. Polled is going to be shown as an uppercase letter and is the dominant trait. Whereas horned is going to be shown as a lowercase recessive trait. So two polled cattle that are homozygous for the polled trait. What does homozygous mean? Homo means the same. So that means that our letters are going to have to be the same. They're either going to have to be both uppercase P's or both lowercase p's. Lowercase means that they have horns, but this right here says that they're polled, so it can't be little lowercase p's. It has to be uppercase p's. So if we were to breed them, and they're both homozygous dominant, then all of their babies are also going to be homozygous dominant, meaning all of them are not going to have horns. All of them are going to be polled. Another example is dwarfism. It happens to some animals. There is a trait for that. So normal sizing cattle is dominant, because we don't see a whole lot of little dwarf cows running around. It'd be pretty cute though. So normal size is dominant, meaning we're going to see it as a capital letter. To dwarfism is the recessive trait. So we have a normal dam, meaning she is going to be dominant. In this example, we're just going to say that she's homozygous dominant. And then we have a sire carrier. Sire means the father. And a carrier means that he is carrying the trait, but it's not being shown. So that means he has one dominant allele, one capital N for normal size. He looks normal on the outside. He's a normal size, but he's carrying and hiding this one little trait for dwarfism. So when they breed, they're still going to have 4 babies that are normal sizes. But there's a 50% chance 2 out of 4 are going to carry for the trait dwarfism. So what happens if both parents are carriers for the trait, for dwarfism? If they're both carriers, that means they have one capital letter, one dominant allele, and then one recessive allele. So when we go through, we have big N. So this is a homozygous dominant baby, meaning it's going to be normal size. Come down here, we have the big N from the dam and a little n from the carrier father, from the sire. Still going to be a normal size because it has that one dominant allele, but it's going to be a carrier because it has that one little n. Same thing for this one right up here, same exact situation. Then we get down here. If they're both carriers, there's a 1 in 4 chance, a 25% chance that they're going to have a baby that's born a dwarf, because you have that little n here from mom and the little n here from dad. So there's a 25% chance that they're going to have a baby that's a dwarf. So this 1 to 2 to 1 ratio means 1 is going to be homozygous dominant. 2 are going to be heterozygous dominant. Hetero meaning not the same. They're going to have one dominant allele and one recessive allele. And then to 1 is going to be homozygous recessive and is going to show the recessive trait of dwarfism. So what you guys are going to do, I need to close out. I'll pull up your worksheet problem. Again, I made it on a PowerPoint because it's just easier for you guys to be able to type up there, especially if we're going to do stuff like Punnett Squares. You have 3 problems that you need to work on working out. Let me know if you guys have any questions on it. Definitely review the PowerPoint if you have any questions on, like, what's the genotype? How do we know what color is dominant? How do we do the Punnett Square? What will the babies look like? So just work on it. Give it your best shot. You guys got this. Hopefully this is just a review for you, but Punnett Squares are very important in biology. If you decide to go to college, you're probably gonna have to take a biology class. That means you're gonna be doing some Punnett Squares. I love you guys. But these Punnett Squares are way easier than the ones you're gonna do in college. So good luck, give it a try. Let me know what questions you guys have, and I'll see y'all.